Dear students, now we will talk about histology of skin and skin derivatives. So, skin, it includes two principal layers. Outer layer is epidermis and inner layer is dermis. Epidermis is epithelium, it's stratified squamous keratinized epithelium which covers surface of skin, and inner layer is dermis. It's fibrous connective tissue. It includes two kinds of connective tissue. Upper layer of dermis, papillary dermis. It includes loose fibrous connective tissue, and reticular dermis. It includes dense fibrous irregular connective tissue. And also under dermis, under skin, we have hypodermis. It's subcutaneous fat which underlies dermis. And in some classifications, we have three layers of dermis, three layers of skin: epidermis, dermis, and hypodermis. In another classifications, we have two layers of skin: epidermis and dermis, and hypodermis or subcutaneous fat is described as distinct layer. And also we have some skin derivatives. They are hair, nails, and glands of skin. There are sebaceous and sweat glands. And we'll begin from structure of skin, and we'll begin from epidermis. So epidermis is outer layer of skin. Here we can see epidermis. It's stratified squamous keratinized epithelium. Under epidermis we have two layers of dermis. It's papillary dermis and reticular dermis. Epidermis, it covers dermis, and between them we have margin. It's basement membrane, which is present in all types of epithelia. So basement membrane, it's border between epidermis and dermis. And here we can see that papillary dermis, outer layer of dermis, it forms papilla, and that's why this contour, this border between epidermis and dermis is wavy. It's wavy line, and it's border between epithelium and underlying connective tissue. Here we can see epidermis, and it's dermis and hypodermis. It's located under the skin, and epidermis, it has some layers. We'll discuss them later. It's stratified squamous keratinized epithelium, and here we have papillary dermis and reticular dermis. Papillary dermis, it's loose fibrous connective tissue, and reticular dermis, it's dense fibrous irregular connective tissue. And here we can see histological slide. It's epidermis. It's stratified squamous keratinized epithelium. Here we can see papillary dermis and reticular dermis. And deeper we have subcutaneous fat or hypodermis. And also here we can see on low magnification histological slide of skin. It's epidermis, the outermost layer. Then deeper we have dermis and thick layer of hypodermis. And we begin from epidermis, outer layer of skin. It's stratified squamous keratinized epithelium, and it has some layers. So here we can see epidermis, and it has layered structure. So let's look at all layers of epidermis. So epidermis, it's epithelium, and all epithelia, they have basement membrane. Here we can see basement membrane, and there is layer of underlying connective tissue. It's beginning from papillary dermis. And on the basement membrane we have the deepest layer of epidermis. It's basal layer or stratum basale. Here we can see deepest layer, stratum basale or basal layer of epidermis. Next layer is stratum spinosum. Here we can see stratum spinosum. Next, stratum granulosum, stratum lucidum, and stratum corneum. There are Latin names, special Latin names for layers of epidermis: stratum basale, spinosum, granulosum, lucidum, and corneum. 5 main layers of epidermis. And here we can see on this histological slide we can distinguish those layers. The deepest layer of cells which are located on the basement membrane, it's stratum basale or basal layer. Next, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. So the deepest layer, the first layer, it's stratum basale. It's located on the basement membrane. Here we can see those cells, and we can see them. They have cuboidal or even columnar shape. Here we can see their shape, and those cells, they have high mitotic activity. Here we can see in a phase of mitosis, those cells, they undergo multiple mitotic divisions, and it provides growth of epidermis and regeneration of epidermis. So stratified epithelia, they are growing from stratum basale, from basal layer, and cells from stratum basale, they are pushed out into superficial layers, and they replace cells of superficial layers, and then old cells on the surface of epidermis, they undergo desquamation. So growth of epidermis begins from stratum basale. There are stem cells and hemistem cells which undergo mitotic divisions, and they provide growth and regeneration of epidermis. And here we can see basal pole of basal cells, and they are attached to the basement membrane. Here we can see basement membrane and connective tissue, and this cell somehow is attached to the basis. So what do provide this? What does provide this? And there are special cell junctions which are called hemidesmosomes. Here we can see a plate of the hemidesmosome, and this plate provides attachment of the cell to basement membrane. There are special intermediate filaments, part of cytoskeleton of the cell, and special substances which fill the space between this plate, and they provide attachment of this plate to basement membrane. And here we can see hemidesmosomes which attach cells to underlying basement membrane. And it's very important because those hemidesmosomes, they attach entire epithelial layer to the underlying basement membrane of epidermis. And here we can see structure of hemidesmosome. So it's plate, special plate, and there are intermediate keratin filaments. They form cytoskeleton of epidermal cells. And here we can see basement membrane. It has lamina lucida and lamina densa. Lucida is a light layer, and lamina densa is dark layer. And there are special integrins, special proteins which provide attachment of this plate to basement membrane. And basement membrane is attached to the collagen fibers of underlying dermis. It's papillary dermis, and there are special collagens which attach this hemidesmosome, and they attach this basement membrane to underlying connective tissue. And here we can see connection between epidermis and underlying dermis. Next layer of epidermis is stratum spinosum. Here we can see, and this layer includes hexagonal, polygonal cells, cells with different shapes, and they are transition from columnar, from cuboidal cells of stratum basale to flattened cells of superficial layers. It's stratum spinosum. Here we can see this layer. You know that cells of stratum basale, they have attachment to the basement membrane via hemidesmosomes, and cells of stratum spinosum are attached to each other and to another cells via desmosomes. Here we can see desmosomes. There are plates into neighboring cells, and keratin filaments, filaments of cytoskeleton, are attached to those plates, and it provides connection between the cells. Here we can see structure of desmosomes. There are keratin filaments. There are special attachment plates and special proteins cadherins which provide connection between neighboring cells. And here we can see numerous desmosomes which form network, and they are attached to the cytoskeleton of all cells. And here we can see that each cell is attached to all neighboring cells, and it provides mechanical properties of the epidermis and attachment of all layers of epidermis and all cells of all layers of epidermis to each other. So desmosomes, they are very important in the epidermis. And why this layer is called stratum spinosum? Because places where cells are attached to each other, they have appearance of spines, of spicules, and that's why cells of stratum spinosum, especially of this layer, they have this appearance. They have spines, spicules on their surface, and it gives the name for this layer, stratum spinosum. And its appearance of stratum spinosum, here we can see cells, they are polygonal, they have multiform appearance, and connections between the cells. There are places where desmosomes are present. Here we can see cells of stratum spinosum, and here also. To those layers, stratum basale and stratum spinosum, they are called germinal layer of epidermis or Malpighian layer of epidermis. It provides growth of epidermis and regeneration. And those desmosomes, they are connected with cytoskeleton of keratinocytes. So keratin filaments, they are located in the cells, in the epidermal cells, and they are attached to the desmosomes, to hemidesmosomes, and it forms a three-dimensional network inside cytoplasm of all keratinocytes. And here we can see keratinocyte and three-dimensional network of keratin filaments. It's cytoskeleton of the cell, and those keratin, it is produced by keratinocytes, and amount of keratin increases, and later it completely replaces cytoplasm of the cells. In superficial layers, keratin cells of epidermis, they become to be keratin plates, postcellular structures, because their cytoplasm is completely filled with keratin. But here cells are alive and they have all essential organelles, and amount of keratin is smaller than in superficial layers. And gradual increase in number of keratin filaments, increase of amount of keratin is called keratinization. It's a replacement of the cytoplasm of epidermal cells with keratin. And superficial layer, stratum corneum, cells of stratum corneum are completely filled with keratin, and they protect underlying cells from damage. And here we can see cytoskeleton of keratin cells in epidermis. Next layer is called stratum granulosum. Look here and look at appearance of these cells. Cells contain granules with special kind of protein. It's complex protein which is called keratohyalin. It's one of the stages of maturation of keratin filaments. There are unmature keratin filaments, unmature inclusions of keratin, and maturation of keratin includes some stages. So in stratum spinosum there are keratin filaments. In stratum granulosum they form granules of keratohyalin, and they are visible in cytoplasm of cells, and it gives the name for this layer. It's stratum granulosum because it contains granules of keratohyalin. Here we can see those granules. And here we can see cells of stratum granulosum, and number of granules gradually increases, and it's bigger in the superficial layers, superficial cells of stratum granulosum. And here we can see granules inside the cells in the stratum granulosum. And here we can see granular cells, granules of keratohyalin. And next layer is stratum lucidum. Stratum lucidum is composed of cells which already are completely filled with progenitor of keratin. It's elidin. Here we can see keratohyalin granules. It's cell of previous layer, and then they undergo some chemical reactions. Some substances also are produced and released here, and next stage it's production of elidin, special substance which fills cytoplasm of the cell, and it replaces nucleus. Nuclei disappear, cells become to be more flattened. Here we can see flattened cell without nucleus, and here we can see cells of the stratum lucidum. They are filled with elidin. Some cells, they contain nuclei, and then nuclei disappear. It's stratum lucidum, fourth layer of epidermis. And in the next layer, cells of stratum lucidum, they are completely filled with mature keratin. It undergoes some chemical reactions and finishes the maturation, and cells become to be more flattened in shape, and keratin plates are developed from pre-existing cells, and those keratin plates, they form stratum corneum. There is different number of layers of keratin plates depending on localization of skin, and those keratin plates, they form stratum corneum. And then some cells of stratum corneum, they undergo desquamation and they are removed from surface of epidermis. It's called desquamation. And here we can see keratin plates. It's stratum corneum of epidermis. Here we can see stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. And also here we can see stratum basale, stratum spinosum, granulosum, lucidum, and corneum, 5 basic layers of epidermis. It's stratum basale, spinosum, granulosum, lucidum, and corneum. And let's compare this type of skin and this. We can see that number of layers was increased, and in this case we can say that keratin layer isn't very thick as here, but stratum lucidum in this case is absent. So we have two different types of structure of epidermis, here and here. In this case, number of layers of keratin layer, stratum corneum, is bigger than in this case. It's thicker, and this type of skin, this type of epidermis, has stratum lucidum, and here stratum lucidum is absent. So we have two types of skin structure, of epidermis structure. We have thick and thin skin. Thick skin has thick, well-developed stratum corneum, and stratum lucidum is present. In thin skin, we haven't stratum lucidum, and we have thin keratin layer, thin stratum corneum. Where those types of skin are present. Thick skin is present on our hands, and it covers our palms and soles, and fingers are covered with this type of skin. And thin skin is more widely distributed. It covers all other regions in our body. And also there are some differences in skin derivatives. Thick skin doesn't have hair. Hairs are absent in thick skin, as well as sebaceous glands, but it has sweat glands. Thin skin, it has sweat glands, sebaceous glands, and hair. So we haven't hair on our palms and soles because those regions are covered with thick skin. And let's compare two kinds of skin under the microscope. It's thick skin. Here we can see stratum basale, spinosum, granulosum, lucidum, and well-developed stratum corneum. And it's thin skin. Stratum basale, spinosum, granulosum, lucidum. Stratum lucidum is absent, and stratum corneum is thinner than in this case. So there are two basic types of skin. Here we can see 5 layers of epidermis: basale, spinosum, granulosum, lucidum, and corneum. And all those cells which we already know, they are called keratinocytes. They are filled with keratin, and they are main cells in our skin, and they are called keratinocytes. They are following stages of maturation beginning from stratum basale till stratum corneum. They transit from one layer to another, and as a result they undergo desquamation. So it's different of keratinocytes, main cells of epidermis. But also epidermis contains another types of cells, and let's look at them. They have another origin, but they also provide very important functions. Here we have melanocyte or pigment cell. It contains granules of melanin which protect our skin from ultraviolet light. Also we have Merkel cell, it's sensory cell. Also we have Langerhans cell, it's macrophage of epidermis. And also we have intraepidermal lymphocytes. Those cells, they provide immune functions, immune defense of epidermis. So, melanocyte. Melanocyte, it contains melanosomes, special organelles, and when our skin is located under sunlight, it causes synthesis of melanin inside melanosomes. Here we can see synthesis of melanin. When we aren't under light, melanosomes are pale, and then they are filled with melanin. So we can see melanin production in melanosomes in melanocyte of skin. And those granules of melanin may be released from melanocyte, and neighboring cells, keratinocytes, they can uptake those melanin granules, and they are called melanophores. They don't produce melanin. Here we can see that keratinocytes, they uptake, they eat melanin, and they don't produce this melanin, but they contain melanin which they have received from neighboring melanocytes. Here we can see keratinocytes which have inclusions of melanin. And here we can see dark and light skin, and in dark skin we can see that there are more numerous granules of melanin which are uptaken by keratinocytes. In light skin we can see that number of melanocytes is almost the same as in this case, but amount of melanin is smaller than in this case. And also melanocytes may be present under epidermis. They may be present in papillary dermis, and groups of melanocytes, they form pigment tissue, connective tissue which contains numerous pigment cells, is called pigment tissue. So here we can see nevus. It's composed of pigment tissue in the papillary dermis of skin. Also we have another types of cells, and next type is Langerhans cell. This cell is located mainly in stratum spinosum, and it provides immune defense of epidermis and our body in general. It provides phagocytosis and also antigen presentation. It receives information about some antigens and sends this information to another immune cells. And here we can see localization of macrophages in epidermis. Also, there are intraepidermal lymphocytes. They are located between keratin cells, and here we can find them. They also provide immune defense. And this cell, Merkel cell, it's sensory cell, it's sensory-epithelial cell, and it sends information to dendrite of sensory neurons, and it reacts to touch, to vibration, so it's one of the kinds of sensory nerve endings of our skin. It's Merkel cell. So we have discussed all cells of epidermis. There are basal cells or cells of stratum basale, stratum spinosum, stratum granulosum, lucidum, and corneum. All those layers are composed of keratinocytes on different stages of their development, and also we have melanocytes, Merkel cells or sensory cells, intraepidermal macrophages or Langerhans cells, and intraepidermal lymphocytes. And here we can see epidermis, and under epidermis we have papillary dermis, which is composed of loose fibrous connective tissue, and reticular dermis, it's dense, fibrous, irregular connective tissue. And now let's go to structure of dermis. So what are the features of the dermis structure? You already know that there is epidermis and papillary dermis and reticular dermis. Border between epidermis and dermis, it's basement membrane, and papillary dermis, it forms folds, it forms papilla, and that's why contour is wavy, and this provides better attachment of epidermis and dermis, and on our fingers, on our palms, it results, this papillary dermis, it results in the formation of fingerprints. They are formed by shape of papillary dermis. It's loose fibrous connective tissue, and reticular dermis, it's dense fibrous irregular connective tissue. And here we can see papillary dermis, papilla, and it's reticular dermis, and under reticular dermis we have hypodermis. And here we can see connection between epidermis and papillary dermis, and you can see here shape of papilla. And here we can see papilla of dermis and its epidermis. Papillary dermis and reticular dermis, it has a lot of collagen fibers, elastic fibers, and they provide elasticity of skin. And here we can see blood supply, blood vessels, which do provide blood supply of skin. We have superficial plexus of the blood vessels, and here we can see artery and vein, and branches from artery, they form networks inside papilla. Here we can see this network, and that's why you may realize that in some cases there are red points on skin, because distinct papilla have own blood supply, and sometimes when we damage our skin, we can see that there are drops of blood because only apical points of papilla were damaged, and we can see bleeding from these damaged blood vessels. That's why we can see where our papilla of our skin are located. So each papilla has own branch of blood vessel, own network of blood supply, and also nerves are entering in each papilla, and they provide innervation of skin. So it's superficial plexus of blood vessels. And papillary dermis, its shape results in the formation of fingerprints. Reticular dermis is composed of dense, fibrous, irregular connective tissue. There are numerous collagen fibers, they provide density of skin, and also elastic fibers which provide elasticity of skin. And those fibers, they have different directions, but some direction may be predominant in some places of our body. And there are longer lines which correspond to the predominant direction of collagen fibers in our skin. Those longer lines should be taken into account during surgical operation. Surgeons should remember about those longer lines. When you cut skin during operation, you should follow those lines. You should follow predominant direction of collagen fibers in the skin. It will result in faster healing of wounds, because if you will cut in another direction, more collagen fibers will be broken, and you should cut less collagen fibers during operations. It will result in faster healing. And under the skin we have hypodermis, deepest layer of skin. In some classifications it's described as third layer of skin, epidermis, dermis and hypodermis. In another classifications we describe only epidermis and dermis, and hypodermis isn't described as a layer of skin. So hypodermis is composed of white adipose tissue, and there are septa which are composed of connective tissue which divide this adipose tissue into lobules. And this adipose tissue, it has very good blood supply, and it provides thermoregulation and also mechanical protection of underlying organs and tissues. And now we are going to blood supply. You know that there is superficial plexus network of blood vessels between reticular dermis and papillary dermis. Also we have deep plexus, deep network of blood supply of skin, and also there are hypodermal blood vessels, and there are anastomosis between those plexuses. So the biggest blood vessels are located under hypodermis. They are given branches to deep plexus of dermis, and branches from deep plexus of dermis are going to superficial plexus, which forms networks inside papilla. And here we can see this structure. Three networks, three levels of localization of blood vessels. And now we are going to skin derivatives, and they are hair, nails, and glands of skin. There are sebaceous glands, sweat glands, and sweat glands may be epicrine and merocrine. So we will begin from sweat glands. Here we can see sweat glands. They produce sweat. They are epithelial structures. They have secretory unit and excretory duct, which is opened on the surface of epidermis. And here we can see structure of sweat glands. Their ducts have stratified cuboidal or sometimes columnar epithelium, and secretory units, they have secretory cells. Usually this epithelium is simple. And here we can see localization of sweat glands and ducts of sweat glands. They are opened on the surface of epidermis. And we have two types of sweat glands. There are merocrine and epicrine sweat glands. They have different mechanism. How do they release their secretory products from the cell? Merocrine cells and merocrine glands, they release secretory products via apical pole without damage of cells, via exocytosis. In epicrine type of secretion and epicrine glands, secretory products are accumulated in the apical pole of cell, and after that apex of cell is removed from the cell. Epicrine glands aren't very numerous. They are located on the arms near external reproductive organs, and they produce sweat which has smell, and it contains different proteins. It contains pheromones. And merocrine glands, they produce sweat which provides mainly thermoregulation, and this sweat, it contains mainly water and also salts and some waste products. This type of sweat, it doesn't have smell. So two types of sweat glands are present. And also third type of glands, it's sebaceous glands. They are related and they have close connection with hair follicles. They are opened in the hair follicles, and they produce sebum, lipid substance, which covers surface of skin and also hairs. Sebaceous glands, they have holocrine type of secretion. Cells of sebaceous glands, they are completely filled with sebum, and then cells die and their membrane is broken and sebum is released into the lumen of secretory unit of sebaceous gland. So here we can see sebaceous gland. Usually their ducts are opened in the follicle of hair. And sebaceous glands are present on thin skin and they are absent on thick skin. And here we can see sebaceous glands, the secretory units and ducts, they are opened in the hair follicle. And here we can see hair follicle, and here we can see hair follicle, its cross section, and sebaceous glands are opened here. And also here we can see sebaceous glands. Its hair follicle, its hair canal, hair is removed, and a sebaceous gland is opened in the hair follicle. And its sebaceous gland and its intact sebum-containing secretory cell and its disintegrating sebum-containing secretory cell, its holocrine type of secretion. Next kind of skin derivative, it's hair, and there are numerous hairs, and here we can see structure of hair follicle. So hair follicles, they have bases which provides growth of hair. It's located in the reticular dermis or even is in the hypodermis. What structures are present there? Here we can see basal membrane of the epidermis and its continued, it forms invagination which goes deeper in the hypodermis. And also we can see here stratum spinosum and stratum basale of epidermis. You remember that they form Malpighian layer, germinal layer of epidermis. Here we can see them. And epidermis also has stratum granulosum, lucidum, corneum, and its thin skin. So only granulosum and corneum layers are present, and lucidum is absent here. 4 layers of epidermis, but only 2 layers are continued into the hair follicle in the invagination. And they form outer hair vagina, inner it corresponds to stratum granulosum and corneum, but its own hair structure. And here we can see papillary dermis and reticular dermis, and also hair follicle. It has hair bag. It's dense connective tissue. It contains a lot of collagen fibers, and they form hair bag. And hair follicle, it has own papilla. It's papilla of hair, and there are blood vessels which provide nutrition of hair, growing hair. And also nerves are coming here. And this region above papilla, it contains growth layers of epidermis, stratum basale, stratum spinosum. So this region is known as hair matrix, and hair begins to grow from the hair matrix, and here we can see growth of the hair. And also in the hair matrix there are melanocytes, and they produce melanin, and those melanin is uptaken by keratinocytes, which are undergoing keratinization. There are 2 kinds of keratin, hard keratin and soft keratin. Epidermis contains soft keratin. Keratinocytes, they contain soft keratin, and hair and nails, they contain hard keratin. And here we can see inner vagina of the hair follicle. And also we have muscle which moves hair. It's musculus erector pili. Here we can see it, and it helps to move muscle. It's called piloerection. When hairs are moving, for example, when you feel cold, those muscles are contracted and it moves your hair. And here we can see structure of hair and its muscle, hair muscle and its hypodermis. So when you feel cold, those muscles are contracted and they move hairs, they move root of hair, hair follicle, and relaxation of this muscle. And here we can see layers which surround hair. Hair has medulla, cortex, and also it has cuticule. Here we can see layers which form hair. And here we can see medulla, cortex, and cuticule, and its epithelial vagina of hair, outer and inner, and its basement membrane and its hair bag. And its hair matrix, its hair papilla, there are melanocytes which produce melanin, and this melanin is uptaken by keratinocytes. They undergo keratinization, and also they uptake keratin. That's why hairs, they have own color. And here you can see hair, it's hair papilla, and there are melanocytes near the hair matrix, and alive keratinocytes near the hair matrix. They undergo multiple mitotic divisions, and then they are moving higher, and here they undergo keratinization. It's medulla of hair, cortex, and cuticule, which covers hair. And here we can see that matrix, it contains melanocytes which produce melanin, and there are two kinds of melanin. It's eumelanin and pheomelanin. Eumelanin, it has brown color, and pheomelanin, it has red or orange color. And when hairs have only pheomelanin, hair has different red and orange shades, ginger hair, and black hair, dark hair, it has a lot of eumelanin, and light hair usually has small amounts of pheomelanin, of eumelanin, and sometimes they contain eumelanin in different amounts. So according to amount of melanin, we have different shades of hair. And when hair becomes to be gray, those keratinocytes, they don't contain melanin. Melanin is produced by, but keratinocytes, they undergo very fast keratinization and they cannot uptake melanin, and that's why gray hair it's denser than usual and it's growing faster than usual hair, and it's a result of bad blood supply in hair papilla. That's why keratinocytes they undergo keratinization faster than usual. And now we are going to structure of another skin derivative. It's nail. There are nails, and they also are derivatives of epidermis, and also some dermal structures are involved in the formation of nails. So here we can see bone of finger, its connective tissue, including connective tissue of dermis, its papillary dermis, and its basal layer of epidermis, and stratum spinosum of dermis. So Malpighian layer, and it's continued into nails, and it forms basis of the nail. And also we have stratum granulosum and corneum, and its top of finger, its cuticule, and here we can see a nail matrix. Here we have region where nails begins to grow, and there are non-keratinized keratinocytes which will form nail, and they are growing here. So it's base of the nail, its nail bed, its eponychium, this region, its hyponychium, and its nail plate. Nail plate is composed of hard melanin, dense melanin, which is related to hard melanin in hair. So here we can see hyponychium, its place of attachment of nail to skin of finger, its nail plate, its eponychium, fold, skin fold, which covers root of nail, its nail matrix, it's located here, place where cells undergo divisions, and in some cases, on some fingers, matrix is visible as hemilun or semilun. It's located here near the basis of nail. Also, we have fold of keratinized layer. It forms cuticule. It's located here, and nail plate. It covers nail bed. And here we can see histological slides of nails, its nail plate, its matrix, its eponychium and hyponychium, its nail bed, its matrix, its finger and its bone of finger, its cuticule, its nail fold, its eponychium, its nail matrix and nail plate. Its nail plate, its nail root, its nail bed, which underlies nail, and its dermis, which underlies nail. And the last question, it's skin innervation. Skin receives nerves which provide sensory function, and there are different sensory structures of skin. So, first of all, there are 3 nerve endings, dendrites of sensory cells. They are common here, and they are branched between epidermal cells, and they directly receive information from outer environment. They usually react to pain, and also they may receive information about another types of sense. Also, we have Merkel cells. They are sensoepithelial cells, and they send information to the dendrites of sensory neurons. There are intraepidermal sensory structures, and also we have sensory structures in the papillary and reticular dermis. In papillary dermis, we have Meissner's bodies or Meissner's corpuscles. They have dendrite. Also, there are glial cells. They cover this dendrite, and also they surround this terminal branch of dendrite. There are glial cells, and they are covered with connective tissue capsule. Those Meissner's bodies are located in papilla of skin. And here we can see Meissner's body or Meissner corpuscle in the papilla under the epidermis. And also we have Pacinian corpuscles. They are present in reticular dermis or in hypodermis. Here we can see dendrite, nerve ending, its glial cell. Also there is inner capsule, outer capsule, and its connective tissue capsule. Inner and outer capsule, they are composed of glial cells. Meissner's bodies and Pacinian bodies or corpuscles, they react to touch, and also they react to tension, pressure, and vibration. So they, together with 3 nerve endings with Merkel's cells, they provide sensory function of skin. Here we can see Pacinian corpuscle and concentric layers of glial cells, and somewhere here we have dendrite ending. So here we can see epidermis, and here we can see nerves, 3 nerve endings. Also, dendrites are going to Merkel cells, intraepidermal structures. Also, we have Meissner's bodies or Meissner's corpuscles. They are present in papillary dermis, and in reticular dermis or in hypodermis we have Pacinian bodies. And also we have Krause bodies. Here we can see Krause body, its dendrite and connective tissue capsule. They are described as thermoreceptors, as well as Ruffini bodies. They have a lot of similarities. Also, we have dendrite, glial cells, and capsule. Pacinian bodies and Ruffini bodies, Krause bodies and Ruffini bodies, they are known as thermoreceptors. So we have discussed structure of epidermis, of dermis, and skin derivatives, and that's all. Thank you for your attention.